we should have a lecture about the whole energy metabolism, but only the overview of it, yes? At the beginning, I will use the PowerPoint, but then I will more draw on the table, yes? For I think that it will be better for you to follow me, yes? And also for those rows, it will be nice to see here the respiratory chain and here the ornithine cycle. So, you know, at the beginning it will be a bit similar what I have already done during the first lecture, yes? Together with Docent Stich, yes? So it means to draw you the energy metabolism, and then after the break I will show you how important it is to learn this, and I will tell you about the specialization of some organs in the energy metabolism, yes? So at the beginning, what is it, energy metabolism? Did all of you know? What is it? Here it's not. So what is it, energy metabolism? We have the whole lecture about it, the whole course about it. So what is it? It has to be something very important.
How to break down food to get energy.
Yes, so how to break the nutrients to gain energy, and then how to synthesize from small compounds the big compounds. Yes, that are used as a storage of energy. Yes, so not only the degradation, but also the synthesis. Yes. Important terms to understand intermediary metabolism is what is it? To understand energy metabolism is to understand what does it mean, and also what does it mean anabolism and catabolism. And what is it? Anabolic pathway, and what is it catabolic pathway? So what does it mean anabolism, anabolic pathway? Which direction in the energy metabolism it is?
To build up
good,
structures in the body.
Yes, so it means from small compounds we build some big compounds, and catabolism and catabolic pathways the other way. So it means from big compounds we do small compounds. Yes. Sometimes we put two words together. For example, you can say that insulin is proteoanabolic hormone. It means that insulin has anabolic effect in metabolism of proteins. Yes. The same, insulin is lipoanabolic hormone. So it has anabolic effect in metabolism of lipids. Yes? Or glucagon is proteocatabolic hormone. So glucagon induces the pathways that degrade proteins. Yes? So therefore you have to understand what does it mean, anabolism and catabolism. Yes? And one hormone can be anabolic in one pathway and catabolic in the other pathway. That's, for example, true for growth hormone. It's proteoanabolic, but in metabolism of lipids, it induces catabolism. Yes? There are some important intermediates in the energy metabolism. What does it mean, intermediate? Does it mean intermediate?
It's not a final product, but also not the
yes.
product.
So it's a product between, it's something between substrate and product. That's the intermediate. The important intermediates of our metabolism are these three. The acetyl coenzyme A. Can you draw its formula? Not the whole formula, only the formula of acetyl. So this is the acetyl coenzyme A. Acetyl is this, and this is coenzyme A. Yes? Here is written S as a sulfur. For here is the bond between carbon and sulfur, and that's important. Yes? Pyruvate How it looks like pyruvate, do you know it? All of you should. So, how many carbon atoms has pyruvate? 3. So this is the formula of pyruvate. So you see that they are very similar, yes? And there is only one reaction between them. The reaction is called pyruvate dehydrogenase reaction, when we transform pyruvate to acetyl coenzyme A. Yes? So these two formulas you have to know. And then what does it mean, NADH?
Energy transport.
Yes. And what does it mean, the energy? It transports, for example, ATP, protons and electrons. So NAD is a transporter of, in general, hydrogen. But hydrogen, it means electron plus proton. This is electron, this is proton. Yes? And inside the electron, we have the energy. And this energy can be released in the respiratory chain. Yes? Or we can put the energy into other compounds. For example, to lactate. When we change pyruvate to lactate, the energy that is inside lactate is higher than the energy inside pyruvate. And the reaction looks like that you let react NADH plus pyruvate and you produce lactate. But NADH, we call that it's a cofactor. It means it helps enzymes to do their action. Yes? So, these 3 intermediates we will need to construct the picture of the energy metabolism. So, where to start to draw the energy metabolism? We can start on the side of nutrients or at the side of the waste products. Yes? So, during the last lecture, I started to draw it here. Where is the respiratory chain? Yes? And I think that all of you now should be able to tell me the whole thing, for you have already seen it. Yes? On the first lecture. So, here is the respiratory chain. What is produced in the respiratory chain? ATP and
Carbon dioxide.
ATP and water. These two things are produced in the respiratory chain. The majority of ATPs are produced in here, in the respiratory chain. What's the substrate for respiratory chain? Somebody else. So what's the substrate for respiratory chain? Protons and electrons. It means this. NADH plus H plus together with one other cofactor, FADH2. Both these cofactors transport protons and electrons into the respiratory chain. How it works, you will learn in one lecture about the energy production inside the mitochondria with Dr. Trnka. Yes? So more information about this, about the respiratory chain, and about the ATP production in a special lecture. Where we can produce these reduced cofactors, these the NADH plus and FADH2. In Krebs cycle. Or citric acid cycle. Citric acid cycle or Krebs cycle. So in this pathway we produce the NADH plus H plus. But not only in this pathway, yes? You remember that there are also other pathways. Other products of the Krebs cycle. CO2. That's the waste product, yes? And also here, in Krebs cycle, we produce some energy. We produce there 1 molecule of GTP. So here in mitochondria, in the respiratory chain, we produce high amounts of energy, but the energy can be produced also in other pathways. For example, 1 molecule of GTP is produced in the Krebs cycle. Here I forget to write one other thing. If you want respiratory chain to work, then you need not only NADH+, but also what? If it's called respiratory chain, oxygen. So here you need oxygen. And from the oxygen you produce water. So here is the oxygen, and therefore we need lungs. And here we have the carbon dioxide as a waste product. What's the substrate for the Krebs cycle? Acetyl coenzyme A. Coenzyme A together with oxaloacetate. If these two compounds, acetyl coenzyme A and oxaloacetate react together, you produce what? Citrate or citric acid. And therefore it's called citric acid cycle. Oxaloacetate has 4 carbon atoms and acetyl coenzyme A has 2. It means how many molecules of carbon dioxide we produce in 1 Krebs cycle. It means from what to here? 2 molecules. Yes? For here we have oxaloacetate, it has 4 carbon atoms. It reacts with 2 carbon atoms, and at the end you have the oxaloacetate. So it means you have to release 2 carbon atoms in the form of carbon dioxide. How to produce acetyl coenzyme A? From pyruvate, that's the one possibility. Pyruvate. How many carbon atoms has pyruvate? 3. The number of carbon atoms is especially important for the metabolism of saccharides. From pyruvate we can produce new glucose, but from acetyl coenzyme A we cannot. Therefore, the number of carbon atoms is so important. Good, so we have the pyruvate. This reaction between pyruvate and acetyl coenzyme A is called pyruvate dehydrogenase reaction, or also PDH, pyruvate dehydrogenase reaction. Also, you can read that it's the oxidative decarboxylation of pyruvate. Yes? It means absolutely the same thing. For here, if you have 3 carbon atoms and here you have 2 carbon atoms, so here you have to release 1 carbon atom in the form of carbon dioxide. And here it's 1 molecule. And if you put this carbon dioxide together with this carbon dioxide, you gain almost all carbon dioxide that is produced in our body. Yes? And therefore, in the test can be that 1/3 of the carbon dioxide that is produced in our body is produced in this reaction. Yes? About 1/3, yes? It's a bit less for carbon dioxide is also released in the metabolism of amino acids. Yes? But the majority is released here and here. How to produce pyruvate? So it means from glucose. And it's not from the glucose itself, but from the glucose 6-phosphate. This reaction is called glycolysis. The opposite reaction is called gluconeogenesis. So glycolysis, and this is the gluconeogenesis. Glucose has 6 carbon atoms. So it means from 1 molecule of glucose, we produce 2 molecules of pyruvate and 2 molecules of acetyl coenzyme A. In the glucose, in the glycolysis, we gain not only pyruvate, but also some energy. In one glycolysis, we gain 2 ATP.
What do we call the reaction from pyruvate to
Gluconeogenesis. Gluconeogenesis. Gluconeogenesis, or in better word, you have it also here. Gluconeogenesis. And this is the glycolysis. Yes, you have special lecture about gluconeogenesis and glycolysis. Yes? How to produce glucose 6-phosphate? Yes, so you take glucose and you phosphorylate it. So that's quite simple. Yes? You take glucose and we phosphorylate it. Unfortunately, in different tissues, we have different enzymes. For example, in beta pancreatic cells and in hepatocytes, we have different enzymes that we have, for example, in muscle cell. Yes? So in the lecture about glycolysis and gluconeogenesis, I will tell you also about this first reaction. For that's very important, for glucose is in your plasma and glucose 6-phosphate is only in your cell. Yes? More information about the glucose 6-phosphate. Glucose 6-phosphate is also important intermediate of the saccharide metabolism. For glucose 6-phosphate can be transformed to other things. If we have a lot of glucose 6-phosphate, then we produce something, especially in the liver, glycogen. What is it, glycogen? A lot of glucose together. Yes? Animals have glycogen, plants have what? Starch. Yes? So it's the same thing as glucose, only the polysaccharide. Yeah? It means a lot of glucose is together. The opposite reaction is called glycogenolysis. It means degradation of glycogen. So this reaction from glucose 6-phosphate to glycogen is the synthesis of glycogen. The opposite reaction is glycogenolysis. Synthesis of glycogen, glycogenolysis. And here, the other possibility. I don't understand.
Cellulose?
Cellulose? No. For you know, in our body we have many other saccharides, not only glucose. But glucose has the central position in its metabolism. So there has to be some pathways how to transform glucose to other saccharides, for example, galactose, or for example, to ribose, or so on. And this pathway is called pentose cycle. You will be able to calculate. More is here. Yes? After the lecture about the Krebs cycle and after the lecture about fatty acids, you can calculate it, yes? But more is here in the Krebs cycle. But in general, if you take fatty acid or glucose, more energy we have here. Yes? For here, we produce not so many reduced cofactors as here. So fatty acid is better. Yes? How to produce fatty acid? From triacylglycerols. And other product is called glycerol. Where we have triacylglycerols, in which tissue? Adipose, especially in adipose tissue, also in the muscle cells, some amount in hepatocytes. Yes? So here we have the other nutrient, triacylglycerol. When we degrade it, we have glycerol and fatty acid. How to use the glycerol? Glycerol has how many carbon atoms? 3. If you do we have here some metabolic pathway where are compounds that have 3 carbon atoms? Here. So glycerol is degraded in glycolysis. There are some reactions that convert glycerol to the intermediate of glycolysis. Or, on the other hand, it can go also to the gluconeogenesis. Yes? For you will learn in the lecture about glycolysis and gluconeogenesis that they are opposite pathways, and the only thing that modulate their activities are the hormones. Yes? And also glycerol is produced from the intermediates of glycolysis. So that was the metabolism of lipids and saccharides. The last thing is here, proteins. And the monomers are called amino acids. So, what to do with the amino acids? Last lecture I said to you that amino acids can be degraded in many pathways and that we can produce many intermediates. Yes? So we can draw that amino acids can be degraded to pyruvate, to acetyl coenzyme A, and also they can go into the Krebs cycle. Yes? And the waste product from all these catabolic pathways is nitrogen in the form of ammonia. And ammonia enters what? Urea cycle or ornithine cycle. And ammonia reacts together with CO2. And at the end we produce what? Urea. And this is the ornithine cycle. So I think that this is the energy metabolism. This you have to know. Unfortunately, you have many other lectures where you will go deep inside every pathway. Only these pathways aren't so many important, yes? For they are very difficult, so we skip a lot of them, yes? For example, the degradation of aromatic amino acids, that's really sweet thing, yes? So these things you have in the books, but you have no lecture about it, and also they won't be in the test, for it's very difficult. But this side you will have to know very good, yes? And also this. So do you have any questions? I forget to write one thing here. Where is pyruvate? Pyruvate can be transformed to lactate. Yes? It's especially important for erythrocytes, for muscle cell. Yes?
Also synthesizing back to amino acids.
Yes, that's good. Here it's also. But there is one difference. We have the number of amino acids in our body. Types. That's the amount of amino acids that are in our proteins, that are encoded in the DNA. But the number of amino acids we have hundreds, yes? Only 20 are encoded in the DNA. But from those 20, we have 12 that we need to eat that are called essential, and then 8 they are 12 that are non-essential, so those we do not, we have amino acids, and we have 12 and 8. 8 we need to eat, they are essential for our body, and 12 are non-essential for our body, so we can synthesize them. So our body has degradation pathway for all 20, but we can synthesize in our body only 12. Yes? So that is sometime in the test that can we degrade, for example, this and this amino acid. So you will remember this is essential, this is non-essential, but it doesn't matter, we can degrade both. But if there is the question, can we synthesize in our body this and this amino acid? Then you have to remember, oh, so alanine is non-essential, we can produce it in our body. But for example, tryptophan is essential, so this amino acid we cannot synthesize in our body, yes? So you do not have to memorize all pathways, you have to only know whether they are essential or non-essential. So these pathways for 20 amino acids, the opposite pathways only for 12 amino acids. So, other questions? If not, then we have the other topic. That's the topic about the specialization of organs in the energy metabolism. Yes? For example, the energy metabolism in liver is absolutely different than the energy metabolism in kidneys. Yes? So, we have 6 organs. Then I want to tell you, it's liver, kidney, muscle, adipose tissue, intestine, and brain. And brain. And before the break, I will do only liver. Then we take a break. So, liver. Unfortunately, here is only one blackboard, so there's no so many places. So, liver. You will learn that liver, that's the key organ for metabolism in our body. If you have patient with liver failure, he has problem everywhere. He has problem in the metabolism of lipids, in the metabolism of proteins, and also in the metabolism of saccharides. Yes? So I will start about the metabolism of saccharides. In liver, all these pathways are active. Yes? Not only catabolic pathways, but also the anabolic pathways. For liver is an important organ for the maintaining of blood glucose level. There is only the normal glucose level is between 3.3 till 5.6, we can say. And liver controls that the glucose level is maintained in this range. Yes? If the glucose level increases, then in liver you start the catabolic pathways. It means if the blood glucose level is high, then in liver you start to degrade glucose to pyruvate and then to acetyl coenzyme A. It goes to the Krebs cycle and you produce energy. But also, if the amount of glucose is so high that the capacity of Krebs cycle is fully used, then in liver you start produce from acetyl coenzyme A fatty acids. So here you see that liver is the organ where we connect the metabolism of saccharides with the metabolism of lipids. And if the value of glucose is high, then the glucose 6-phosphate is also transformed to glycogen. So you see the three ways how liver can decrease the blood glucose level. If you have opposite problem, it means when we have low blood glucose level, then in the liver you switch the metabolism and you activate the opposite pathways. So here we have the glycogen, and glycogen is degraded to glucose 6-phosphate. And then here, the glucose 6-phosphate is transformed to glucose, and glucose is released to your blood. But if you have no glycogen in liver, then here we have other pathway how to produce new glucose. You take, for example, here some amino acids, or here you take some glycerol, or here you take the pyruvate, and in the process of gluconeogenesis, you produce new glucose 6-phosphate and new glucose. And to have energy for this process, in liver you degrade fatty acids in the beta oxidation. You produce acetyl coenzyme A that is degraded and you produce energy. And this energy is then used here in the Krebs cycle, in the gluconeogenesis. So in liver, all these pathways are active. Only they can switch according to the body demands. It means if you have, for example, a lot of insulin, you degrade glucose and you produce new fatty acids. If you have a lot of glucagon, that's other hormone that regulates the energy metabolism, you degrade glycogen, produce new glucose, and you degrade fatty acids. Yes? So here you see the role of liver in the metabolism of saccharides. Metabolism of lipids is closely connected to the metabolism of saccharides. So you see that you can degrade glucose and you can produce new fatty acids. On the other hand, if you degrade fatty acids, you can produce energy for the synthesis of new glucose. Yes? But also in your liver, you can take this acetyl coenzyme A and to produce some compounds, and especially they are produced from the fatty acids. So if you have no food, so you need to produce new glucose. But the problem is that the new glucose can be produced only from amino acids, from pyruvate and glycerol. But we have a lot of fatty acids. So therefore, in the liver, if we have no food, the fatty acids are degraded. We produce the energy, but in addition, we take the acetyl coenzyme A and we produce compounds that are called ketone bodies. And the synthesis of ketone bodies we can find only in liver and only if you have no food, yes? Or there are some other pathologic conditions, for example, type 1 diabetes, where we have also a lot of ketone bodies. So this is the special pathway in liver, the production of ketone bodies. No other organ can produce ketone bodies. Yes? Ketone bodies, we put them in the same group as the triacylglycerols, so in the big group of lipids. But triacylglycerols are nonpolar and ketone bodies are polar, yes? So they are not the proper lipids, yes? So that was the metabolism of lipids in the liver. So here it's connected with the metabolism of saccharides and the unique pathway here, production of ketone bodies. And in the metabolism of proteins and amino acids, all plasma proteins are produced in liver. Only antibodies are produced in B lymphocytes. But all other, it means, for example, albumin, C-reactive protein, coagulation factors, all are produced in the liver. Yes? The normal value of blood proteins is about 70 grams. Yes? So the liver has to produce a lot of proteins every day. In metabolism of amino acids, all amino acids are degraded in liver. Yes? Only one group of amino acids is degraded in muscle cell, but all other groups are degraded in liver. Yes? And also, here in liver, we have the ornithine cycle or the urea cycle. And about 95% of the total capacity of ornithine cycle we have in liver. The other 5% are in the other cells in our body. Yes? So in the metabolism of proteins and amino acids, almost all plasma proteins are produced in liver. Almost all amino acids are degraded in liver. And also the non-essential are synthesized in liver. And the majority of urea is produced here in liver. So the worst question about the energy metabolism is the role of liver in energy metabolism. For you have to know everything. Yes? For all pathways are localized here. So do you have any question? That's so boring or so difficult? What? The first. So boring. Lot of informations. I know that you know, but this should be the introductory lecture to all the other. Yes? For you have the lecture about glycolysis and gluconeogenesis. You have special lecture about degradation of amino acids. You have special lecture about Krebs cycle. You have special lecture about respiratory chain. We have special lecture about degradation of lipids. Yes? But the first lecture should be everything together. Yes? And then during the lecture, the other lectures, the teacher should tell you, yes, now we will do this. Now we will do this. Yes? But you have to know the whole context. Yes? How they are connected together. And that also during the last lecture, the teacher should draw you this, you to understand it in general. Yes? So some questions?
Just how the glycolysis, how it depends on the Krebs cycle, how much it depends on the Krebs cycle too.
The problem is that if you stop the Krebs cycle, then all these pathways that feed the Krebs cycle have to stop. Yes? So that's the connection. If the Krebs cycle isn't working, then beta oxidation also, glycolysis also. And the Krebs cycle, together with respiratory chain, produce a lot of energy. If you have a lot of energy, you will stop the glycolysis and also the beta oxidation. So all these pathways have to cooperate, yes? So you have to produce the precise amount of substrate. For this substrate is the product, the precise amount of product. For this product is substrate for another pathway. Yes? And the cell doesn't want something to accumulate, yes? It likes the equilibrium in everything.
Because it's the pyruvate who goes into Krebs cycle, right?
Mm-hmm, yes, that's the chemical connection. Yeah. Yes, but then you will learn about the regulation, and you will learn that the regulatory enzyme of glycolysis depends on the amount of reduced cofactors and on the amount of ATP. And they are produced here, reduced cofactors here, and ATP here. Yes? So that's the metabolic connection between these two pathways. Yes? This is the direct, but the regulation is here, together with the amount of pyruvate. So there is no question, so we have the break. So we will continue with kidney. So why we need kidneys? Why we need kidneys?
To get the waste products?
Yes. To excrete waste products from our body, especially those waste products that are soluble in water. Yes, it means polar waste products. You know that every day we produce about 2 liters of urine, and to produce it, the kidneys need a lot of energy. So all pathways where the kidneys can gain energy are very active in kidneys. Yes. So it means they can degrade fatty acids, they can degrade glucose, and also they can degrade amino acids. Very often you will read in the books that their main substrate are amino acids. Yes, that's not true, but the role of the metabolism of amino acids is very important in kidneys. If we have some problem with ornithine cycle, only in kidneys we can excrete the ammonia from our body. And there are some conditions when we have to suppress the ornithine cycle, for example during acidosis. And then the amount of ammonia excreted in kidneys is higher than normal. Yes? So therefore you will read a lot of things about the metabolism of glutamine and glutamate in your kidneys. You will learn that kidneys need a lot of glutamine. But not only glutamine, yes, it's also the glucose and also fatty acids and also ketone bodies, yes? But it's true that the glutamine has important position in kidney metabolism. For glutamine can be degraded to glutamate, and glutamate can be degraded to alpha-ketoglutarate, and alpha-ketoglutarate is intermediate of Krebs cycle. So if kidney takes glutamine, it has also energy here in Krebs cycle. But more important is that the ammonia can be excreted directly to the urine and not in the liver to be transformed to urea. So that's important. That's about kidneys. Other important position in intermediary metabolism is that this pathway, it means the gluconeogenesis, is localized not only in liver, but also in kidneys. Yes, so about 10% of the total gluconeogenesis capacity is localized in kidneys. So that's important. About 85% in liver, about 10% in kidneys. Yes? So if you have kidney impairment, you can suffer from hypoglycemia. It means from low amount of glucose in your body. Yes? For you are missing 10% of gluconeogenesis. But of course, if you have liver problem, you suffer more often from hypoglycemia. Yes? Than if you have problem in kidneys. Then we have a muscle cell. Why we need muscle cell? Why we need your rhabdomyocyte? For the muscle contraction. Yes? And the muscle contraction is only the transformation of chemical energy to the mechanical energy. So if your muscles are working, they need a lot of energy. And the problem is that in one moment you can sit, and in the other moment you can run. Yes? So in muscle there has to be a lot of molecules that are able to store the energy. Do you know some of them?
Glycogen.
Glycogen. We have the muscle glycogen. Yes, that's important for the fast action. Other molecules. So glycogen is here. Inside rhabdomyocytes we have also triacylglycerols. Yes, triacylglycerols aren't only in your adipose cells, but also in rhabdomyocytes. And also, for the fast action, we have there the phosphocreatinine. Yes, so that's the molecule that can be directly transformed to the ATP. Yes, or it's better to say, if you break the molecule between creatine and phosphate, this phosphate you can put on the ADP to produce the ATP. Yes? More about the energy metabolism of muscle cell you will learn in the second year, where you have special lecture about the energy metabolism of muscle cell. Yes? For it's very closely connected with the contraction. But one other important thing about the metabolism of muscle cell is that I have already said that all amino acids are degraded in liver, but there's one exception, that are the branched amino acids. Yes, the abbreviation is BCAA or only BAA, branched chain amino acids. Do you know them? There are 3, so the first is so these 3 only these three are branched chain amino acids. And degradation of these amino acids starts in muscle cell. You change them to some intermediates, and then these intermediates go to plasma, and they are degraded in liver. But the first phase of their degradation is localized in muscle cell. And that's important, for you can, in some pathologic condition, you can calculate the ratio between these amino acids that are normally degraded in the muscle cell and with all other acids that are degraded in liver. Especially we calculate something that's called Fisher index. That's the ratio between these amino acids and the aromatic amino acids. And the Fisher index is very useful if you have the patient with liver impairment. It's the easy way how to find out how severe is the problem. So you will compare the concentration of these amino acids with the concentration of aromatic acids that are normally degraded in liver. And if the amount of aromatic acid is very high, you see the problem is very severe. Yes? So then the Fisher index is lower than normal. But that you will learn in the hepatology lecture, not now. So that's the second important thing about the energy metabolism of muscle cell, degradation of these three amino acids. Then we have adipose tissue. So, what's the role of adipose tissue in our body? What's the role of adipose tissue?
Insulator.
Yes, isolator. That's important for biochemistry. But that's important for, then you do not lose so many heat.
Storage of energy.
So it's the storage of energy. Yes? We have the glycogen can feed you only for 1 day. Fatty acids in adipose tissue can feed you for 40 days. Yes? So here you see how important it is to have a lot of fatty acids, but only in the case that you will starve. Yes? So we have two conditions, or you will learn that the adipose tissue has many other functions. It's very active as an endocrine organ. Yes, we have the whole group of hormones that are called adipokines that are produced in adipose tissue. Yes, but about this you will learn also in the 2nd grade. Yes, when you will learn about the hormones. But for now, the first role of adipose tissue is to store triacylglycerols, and the second role is to release triacylglycerols. And to do this,
What's the first one? Sorry.
storage of triacylglycerols. So it means to have energy, and here to release the energy. And we have there two enzymes to do this. The first is lipoprotein lipase, LPL. And the other enzyme is hormone-sensitive lipase. For the first function, you need the first enzyme, the lipoprotein lipase. This enzyme is localized on your endothelial cells. Yes? And the lipoprotein lipase degrades triacylglycerols that are in your plasma. Lipoprotein lipase degrades these triacylglycerols. You release the fatty acids. And then the adipocyte can take these fatty acids. And inside its cytoplasm, it can produce new triacylglycerols. Yes, so this lipoprotein lipase is on the surface of the endothelial cells, and it degrades the triacylglycerols that are in your plasma. Yes? The other enzyme, the hormone-sensitive lipase, is an enzyme that is localized inside adipocytes. It means inside the cells of adipose tissue. And it's active only when we need to release energy.
What do you call it?
What a hormone-sensitive lipase. Yes? This enzyme is regulated by hormones for its hormone-sensitive lipase. But the problem is that also this enzyme is regulated by hormones. Yes? So all of them are regulated by hormones, but only the second has it in its own name. Yes? And the function of the hormone-sensitive lipase is to degrade the triacylglycerols that are inside the cell and then to release them into your blood plasma. Yes? And then these fatty acids can go to liver, where they can be degraded. So you see that here and here, in both cases we need a lipase, but the name is different and the function is absolutely different. Yes? So please do not mix these two lipases. Yes? This is the anabolic lipase. It's used when you have a lot of insulin in your body. This other lipase is catabolic, and it's active when we have lack of insulin in our body, when we have a lot of glucagon in our body. So that's about the role of adipose tissue in energy metabolism.
Did that produce leptin as well?
Yes, that leptin is one of the adipokines. So adipose tissue can produce, for example, leptin, but it can also produce, for example, tumor necrosis factor alpha that is regulating the inflammation in the body. It can also produce the resistin that decreases the insulin sensitivity. So now we know that adipose tissue can produce 20, 30, 40 different hormones. Yes? So it's very difficult, but not for the first grade. But of course that during, for example, the internal medicine exam, you will have to know it. For it has a very important role in the diabetes type 2, these hormones. Not here. The other is intestine. So why we need intestine? To digest and to absorb nutrients. Yes? But it was discovered that in the enterocyte, that's the cell of intestine, the nutrients are transformed. Yes? If you look what amino acids we eat, and then if you measure the amount of amino acids in your circulation, you will discover that it's absolutely different. Yes? And if you measure the same in the portal vein, that's the vein between intestine and liver, you will discover that it's also different. So we know that enterocytes change the amount of amino acids. From some amino acids, they produce glucose. From some amino acids, they produce other amino acids. And some amino acids then they take and degrade in the Krebs cycle. Yes? More information you will have in the metabolism of digestive system in the second grade. Yes? But only remember that in intestine, you can change many things, yes? Not everything what you eat has to go to liver, but something is also transformed in the intestine. And other important thing for energy metabolism is that about 5% of total gluconeogenesis is localized in enterocytes. So at the end, if we have the gluconeogenesis, 85% we have in liver, 10% in kidneys, and 5% in enterocytes. And do you know how we find out tissues that can do gluconeogenesis? For all these tissues have some enzyme. And for example, a rhabdomyocyte doesn't have this enzyme. And therefore, these tissues can do gluconeogenesis, and muscle cell cannot do gluconeogenesis. So if you want to find out, it's here. Is the enzyme that's called glucose 6-phosphatase. That's the enzyme that takes phosphate from the glucose 6-phosphate and it produces glucose. In these organs, we have this enzyme, but for example, in muscle or in brain, there is no glucose 6-phosphatase. Yes? So that's the important enzyme for the gluconeogenesis, this last enzyme. And then the last, we have the brain. So why we need the brain?
Think.
Yes. Not to be apes. You know that brain consumes about 20% of total energy expenditure of human body. Yeah? About 20% of oxygen is consumed there. So you know that brain needs to have a lot of energy. What do you think is important for brain? Which nutrient is important for brain? This. And is there any other?
Ketones.
Then ketones. So those are here, ketone bodies. So for brain energy metabolism, we use these two compounds, glucose and ketone bodies. Under normal condition, it's only glucose. About 120 grams of glucose we need every day, only for brain. But in adopted starvation, about 50% of energy expenditure can be covered by ketone bodies. Yes? So that's important for the metabolism of brain, the glucose, and the ketone bodies. The precise metabolism is more difficult. For in brain we have two types of cells. We have there are the neurons, and then we there have the astrocytes. And you will learn in the fourth course that the astrocytes take glucose, they degrade it to pyruvate, then they transform pyruvate to lactate, and lactate they give to the neuron. Yes? So the brain needs glucose, but neuron needs lactate. Stops the York-Krebs cycle. Yes? The precise mechanism is that if you have a lot of ammonia, the body wants to destroy it. But if the urea cycle isn't working, Then it takes some intermediates of Krebs cycle, for example, alpha-ketoglutarate. And if you have alpha-ketoglutarate together with ammonia, you produce glutamate. And from glutamate, you can produce glutamine. But if you take all alpha-ketoglutarate from Krebs cycle, you will stop the Krebs cycle. And for in brain, Krebs cycle is very active. Therefore, the first signs of high amount of ammonia in your blood you can find in your brain. You have the brain swelling, and at the end you have brain or you have coma. Yes, we call it liver coma. For the problem is in your liver, but the first organ where you will find it is your brain. Yes, so therefore it's liver coma. So that was about brain. So 4 minutes. Inter-organ metabolic pathways. What does it mean, inter-organ metabolic pathway? That's quite simple. You have 2 organs. And you want to find some connection between them. We have many inter-organ metabolic pathways. For example, in liver, we transform glucose to acetyl coenzyme A. In liver, we transform acetyl coenzyme A to fatty acids. And then these fatty acids go to your adipose tissue, where the lipoprotein or fatty acids are transformed to triacylglycerols. And these triacylglycerols are degraded by lipoprotein lipase, and they are stored in adipose tissue. That's one inter-organ metabolic pathway. Or in muscle cell, you degrade, for example, valine. Yes? One intermediate is released into your blood. Then this intermediate is caught in liver. And in liver, you change this intermediate of valine in Krebs cycle, and you produce energy. So that's the inter-organ pathway between muscle cell and between liver. Yes? Here I had some other pathways, but we will do it once more. Here, these 2, the Cori cycle and the glucosalanin cycle, yes? For we have lack of time, I will tell you about them in the lecture about glycolysis and gluconeogenesis. Yes, so I will skip it. For it's closely, that's not the end. We have deregulation. So I will skip it. I will tell you about it in the lecture about glycolysis and gluconeogenesis. And the last topic is the hormone regulation. So take some color. And for example, the red color will be insulin. So, the regulation of energy metabolism. On one side we have insulin, on the other side we have hormones that we call contra-regular hormones. Yes? Contra-regular hormones. Insulin is only one soldier, and we have 4 contra-regular hormones. So it means one insulin can fight with 4 other hormones. Yes? The contra-regular hormones are, for example, glucagon. It's also the growth hormone. It's cortisol. Yes? You will learn about them more in other lectures. But today, I will show you the effect of insulin and the effect of glucagon. So, insulin. Insulin is an anabolic hormone. So it wants to produce big compounds. So that's the first definition. Insulin is an anabolic hormone. But the other definition is also important. For insulin wants to do everything to decrease blood glucose level. And if you learn these two definitions, you will understand everything. For the first, it means it's anabolic hormone. Decreases blood glucose. Insulin is anabolic hormone, so it means from acetyl coenzyme A, it will produce fatty acid. From fatty acid, it will produce triacylglycerols, and it will activate the lipoprotein lipase. In this metabolism of proteins and amino acids, insulin takes the intermediates and it will produce 12 amino acids, those that are non-essential. And also from amino acids, it will produce proteins. But the second definition says, insulin wants to decrease blood glucose level. So here, we have the glucose in your blood. And how to decrease the blood glucose level? To degrade the glucose. And also to transform glucose to glycogen. And also to transform glucose to other saccharides. So this is the effect of insulin. So it's proteoanabolic hormone. It produces new amino acids. It's anabolic in metabolism of lipids. It produces new triacylglycerols. It's anabolic here, for it produces glycogen, but here it's catabolic. Yes? So you can say it's proteoanabolic hormone, lipoanabolic hormone, but here it's more difficult. In some pathway it's catabolic, means in this, and in this pathway it's anabolic. And if you look at glucagon, it has opposite effect. Everywhere it's opposite, yes? But in the books you will learn that, for example, the glucagon, it's not so effective here. That here is more effective, for example, cortisol. But for now it's not very important. Learn that we have insulin and we have glucagon. They fight together. If you have more insulin, it looks like here. If you have more glucagon, or in general more contrarregulatory hormones, we have the opposite effect. So any questions? So what you will do,
Wasn't glucagon that have to be forward to beat insulin? You said insulin could beat for
yes.
something.
You know, the relationship between insulin and glucagon is very difficult. For insulin is produced in beta cell, glucagon is produced in alpha cell of pancreas. And there are some relationship that if you will produce insulin, you will also produce some amount of glucagon. If you produce glucagon, you will produce also some amount of insulin, yes? So more about this relationship you will have in the second grade, yes? The important is the ratio, not the precise level.
But yeah, it depends on which state you're in.
Yes.
Like if you're hungry or you're...
Yes,
or
hungry, glucagon, after meal, insulin.
Yeah, exactly.
So another question, and what's the first thing after the lecture? To evaluate it.
Evaluate it.
So if there are no other questions, then see you on Thursday.